Hello, everyone. I'm Alex Ascher. I'm a postdoc at Quogue Bay Conservancy and Hui. I'm going to talk about some ALI-funded research I did, along with it was led by Carolyn Teepolt and Jesus Pineda at Hui. We're interested in lobster larval trophic dynamics within convergence zones. So the larval period is punctuated by a need for growth, and growth requires food. A lobster larvae from stage 1 to stage 4, over their roughly 30 days development, increase their mass by almost 20 times, actually a little bit more than 20 times over those 30 days. And that type of rapid growth means you need a large supply of high quality food. However, the planktonic prey that larvae rely upon are very patchily distributed within the surface oceans. And the mean density of prey that's available to them is likely not enough to support the type of rapid growth that we see and that they need. So to meet their energetic needs and their growth needs, they may need to rely upon aggregations and dense abundances of prey that are otherwise not available in average situations. So we have convergence zones, which is kind of like if you had a buffet, but in the water. You can see kind of over here in this image, the line of buoyant algae going across here is an example of the convergence zone. Convergence zones are basically like these ephemeral small-scale features in the surface oceans. They can be produced in a variety of ways. They take place in a lot of times at interfaces, like at tidal fronts or river discharge. And they're interesting and important little microhabitats because they concentrate buoyant material such as algae, but also a lot of plankton and zooplankton prey. Here's kind of like a cross-sectional view to give a better understanding of kind of how convergences form and why they aggregate zooplankton. So in this example, you can see how these patterns of internal like upwelling and downwelling causes convergence to form and aggregate along the surface here. And it aggregates a lot of zooplankton. And it's been shown in larval fish literature that larval fish utilize these as important feeding grounds and as microhabitats. And kind of anecdotally in observations over the years, people have also seen lobster larvae associating with these convergence zones. But there hasn't been a pointed study of this. And so we became interested in this question. Do convergence zones impact the trophic dynamics of lobster larvae? Are lobster larvae also utilizing these microhabitats as important feeding grounds? And so during 2021, a sort of 16 potential convergence zones were identified within the Gulf of Maine. Of those 16, when they were sampled, 11 actually had active convergence sites that while sampling was being conducted. So we'll focus on those 11 sites for the rest of this talk. At those 11 convergence zones, there are plankton tows conducted through the convergences and then also outside, but just alongside the convergences. And each of those sites, the abundance and identity of the zooplankton were quantified both inside and outside, and then also the lobster larvae in their stage. And then I took a combination of microscopy and metabarcoding to look at the diets of lobster larvae, both inside and outside those convergence zones. Of course, we only found enough stage 1s and stage 4s just to do a real analysis, so just forget the stage 2s and stage 3s. It's just all stage 1s and stage 4s. The other two don't exist. I'm going to be showing a variety of graphs from trying to look at associations between stage 1s and stage 4s and the convergence zones and their prey, starting out with these bar charts where we're looking at prey or stage 1 prey density over here and number per meter squared, stage 1 density and stage 4 density, and then on the x-axis are the convergence zones, the site numbers. So don't worry that we're missing 8. That's because that wasn't a real convergence zone. Broadly, what we're seeing is that the prey are associating with those convergences. We see peaks of prey inside the convergence zones, so they're typically more abundant within the convergences. Stage 1s and stage 4s, we typically see peaks that are inside those convergence zones as well, but it's really variable. So it's hard to draw like really tight statistical associations, especially with just 11 sites. So don't worry about stats. You're not going to see any of them. We're looking at patterns and we're squinting our eyes at graphs. But what we see that's really interesting is when we do see these really dramatic peaks, especially for stage 1s inside convergences, they line up really closely with abundances in prey. There are also the outliers. Convergence number 9, there's a big peak in prey both inside and outside, and yet no peak with stage 1s. Convergence number 2, the stage 1s kind of associated equally in both areas, and the stage 4s are kind of doing their own thing. They're peaking in different areas, but not in relation to the prey. So first off, I want to look at this just relating stage 1 and stage 4 abundance to prey density, both inside and outside the convergence zones. In these graphs, so stage 1 density and stage 4 density, and the numbers are the convergence zones. Those are going to act as my points so that we can kind of follow some of these convergences. And what we see is there is a significant trend towards positive trend for the stage 1s inside the convergences, whereas you see more prey inside convergences, you also see more stage 1s. Not outside the convergences, and not in the stage 4s one way or the other. We're still seeing convergence number 9 pop out as a little bit of an outlier, but potentially there's a good, easy explanation and good reason for this, which is that the abundance of stage 1s inside or outside convergences depends on a whole lot of different factors. And for one, you're not going to see a peak in stage 1 abundance if there isn't larval supply when that convergence forms. So that gives me an idea for how to kind of plot these densities and abundances out to get an idea of how stage 1s and stage 4s are aggregating or not aggregating around convergence zones, where to take care of this issue of larval supply having an impact on their abundance and density inside and by location, you can plot just the total number of larvae in the sampling area. So in this little schematic, this is our sampling area. The red is a convergence, the blue is not a convergence. If you take all of the larvae, total density outside, and plot that on your x-axis, and then the y-axis is your specific inside and outside densities. If they are randomly or uniformly distributed around these convergences, you should see about equal slopes. If they're aggregated, you should see those slopes diverge. And if they're really aggregated, you should see more of a divergence. And when we plot this for our prey, our stage 1s and stage 4s, we see some of these patterns. So prey are associating and aggregating within those convergence zones. Stage 1s, now we kind of see something where largely there just is not a whole lot of supply of the stage 1s. But when there is a supply of stage 1s and it matches up with times where we have a large density of prey, they really fit this line where they're almost completely only within the convergence zones. And for stage 4s, again, kind of doing their own thing. They sort of aggregate within the convergence zones. It doesn't really seem related to prey, and it's not really as strong as what we see in the stage 1s. The stage 1s, however, like I said, we see dramatic peaks in convergences, and they associate with prey. And so the next part is understanding and getting interested in this idea of, well, are they eating different things inside or outside? So again, I use two different methods here. I have my metabarcoding and I have my microscopy. And the table is showing my sample sizes for each. In the metabarcoding, I only looked at stage 1s and 4s inside just due to sampling and money. And then microscopy is where we have a little bit of a larger data set and where we'll do some comparisons inside and outside. For the metabarcoding data set, I used two different primer sets, COI and 18S. COI performed a lot better than the 18S. We're going to focus just on the COI is what I'm going to demonstrate today. In the future, we have finally funding that was meant to come through like a year ago, finally got a green lit, and we have funding to do the 18S and redo it, but with a blocking protocol that I've had success with in the past, and potentially increase our sample size there as well. So forthcoming. And then alongside the microscopy, we also looked at the gut fullness of the larvae and also the length of the larvae. Are we going? There we go. Okay, cool. So here's some of the microscopy diet data set. A little bit of I've cut it short a little bit so that it's not too overwhelming. The way to read these graphs, when I do my microscopy and my visual analysis of the gut contents, I will, for an individual, I will rank presence or absence of different prey types, depending on what I'm able to identify. I'll identify it to the lowest taxonomic degree I can, and then at each of the corresponding higher degrees. So in other words, if you have a Acartia, I will say the genus Acartia is present, so is the family Acartiidae, so is the order Calanoida, the class Copepoda, and the phylum Arthropoda. I don't know why I started the genus as a word. I wasn't going to remember all the right ones. Then I plot them by the proportion of the individuals which had that specific prey item in there. Don't worry too much about bars not reaching 1. That's because sometimes guts are empty, or sometimes they can't identify as many down to like lower levels. But this gives us a broad way to just sort of investigate the diet. First, what we're seeing is, with microscopy at least, stage 1s don't appear to have as broad a diet as the stage 4s on the bottom here. So more prey types in the stage 4s. And then I would say if we're characterizing diet inside and outside, roughly similar for both stage 1s and stage 4s. I don't really see enough to say that there's a big difference. There may be some small differences, like over here, this is Malacostraca for stage 1s, maybe more inside. But with the sample size we have, I wouldn't really call that a significant difference. So I see this as being roughly same diet inside and outside. We do see the dominant prey types. So copepods are most dominant for stage 1s with a little bit of malacostracan, which is things like decapods, amphipods, isopods. Those larger malacostracans are much more important for the stage 4s. And then also interestingly, their secondary and most important diet type is insects, which are extremely abundant within the Newston layer, and especially within those convergence zones. When we look at our metabarcoding data, again, this is just larvae from inside. So we're just looking at expanding our knowledge of diet here for the stage 1s and stage 4s. So the blue bars are stage 1s, the brown bars are stage 4s. We get some interesting additional context here. One is the stage 1s now are showing a much broader diet than I was able to determine with microscopy. And there's some specific prey types which are really interesting. One is fish. So red hake and monkfish showed up in the sequencing, both of those. They also showed up in our plankton nets in a great abundance, and eggs and larvae. So with the metabarcoding, we're finding some potential prey types that we can't really see through microscopy. Eggs, and probably mostly eggs for the stage 1s, could be important and could be really nice little tidy bundles of lipids and fats and proteins and whatever. Additionally, malacostracans didn't appear to be all that important for the stage 1s in the microscopy, but now we see a pretty good peak for malacostracans for the stage 1s, mostly related to Hemigrapsis sanguineus, the invasive Asian shore crab, and Meganictiphines norvegica, the northern krill, being present in pretty high abundances within the stage 1s. When we translate all of this back to convergences and the use of convergences as feeding zones, while the diets of stage 1s and stage 4s inside and outside are roughly similar, the abundance of preferred prey types is really different. So this is now showing prey type and the density of prey type summed across convergences inside and outside. And those preferred prey types, the insects, copepods, and malacostracans, those are really significantly more abundant and more dense within convergence zones. So if you think about the stage 1 and stage 4s that are resident within the convergence zones, they're probably realizing a much higher feeding rate and expending less energy to fill their guts. Now, you might expect that to translate to some differences in either size or gut fullness, but at least for size, you'd be wrong. The stage 1s and stage 4s, inside and outside, their lengths were almost exactly the same. For gut fullness, stage 4s at least, ooh, I lost my pointer. Stage 4s at least, you don't really have a lot to see. Stage 1s, there was an interesting pattern. Inside convergence zones, so I rank gut fullness as 0 to 25, 50, 75, 100% full. Inside convergence zones, stage 1s have a really normal distribution to their pattern of gut fullness. Outside convergence zones, it appears it begins normal, and then you actually see a peak at 100% gut fullness. So maybe opposite what you would expect based on what we just talked about with the abundance of prey difference. But if we dive a little bit deeper, this pattern is actually a little bit more interesting and pops out an interesting relationship here, where when we take size and we plot that on the x-axis for the stage 1s against gut fullness, what we see is that outside of convergence zones, size is correlated with gut fullness. So the largest stage 1s outside of convergence zones are the ones who are able to fill their guts. While inside the convergence zones, there is no relationship between size and the ability to fill your gut. It relies on something else. And so what I interpret this as is that outside the convergence zones, that is a more strongly selective environment where only more fit, adapted, larger individuals are able to feed efficiently. Whereas inside the convergence zones, the abundance of prey means that that's relatively low energetically costly and then easier to fill their guts. If we try to wrap this all up in a nice little bow, what we're seeing is that there is patterns of aggregation for both stage 1s and stage 4s within the convergence zones, more so for the stage 1s. More importantly, when we see the supply of stage 1s match up with abundances of prey when convergence zones form, we see huge dramatic peaks in stage 1 abundance within those convergence zones that you don't really see otherwise, well beyond the background amount of larvae. And we also see that preferred prey are aggregated within those convergence zones and likely support higher feeding rates. The stage 4s, also appear somewhat aggregated within the convergence zones, but they're kind of doing their own thing and it doesn't appear related to prey. So either they're using that microhabitat for a different reason, or their ability to, their stronger swimming ability means they can kind of choose whether they're resident within or outside more actively. All right, with that, I'd like to thank my funding from ALI, as well as Sarah Zeidema and Sarah Shapira at Hui, who did a lot of the prep for these samples, and I'll be happy to take any questions.
Thank you. Anyone have any questions?
Alex, really nice talk. That was really interesting. I'm curious, you're looking at size differences of things that you collect in the actual convergence zone. But if they're getting a bump from feeding there, like say the stage 4s, wouldn't you see that translate into how big they are when they molt to stage 5? And I was just wondering if you guys kept any of them to see how that turned out.
Jesus kept some of them for some starvation experiments. I don't know if they kept them for molting. The size differences that I was expecting, so these are also ephemeral. These are lasting usually like hours. So to give a scale idea, these are maybe tens-ish of meters across and like hundreds-ish of meters long. And like I said, form usually for a couple hours. So I wouldn't really expect size differences to be based on growth rates inside or outside. I would expect more, like I was saying, to maybe selective pressure to be the thing that matters, or size relating to swimming ability and your ability to remain resident within the convergences.
Thank you.
Thank you for the talk. That was really interesting. I've had the pleasure / displeasure of trying to sample convergence zones, and I know how fickle they are. So I'd like to know your methods for how you were sampling within the zone, and then how far away you're out of convergence zones were.
Yeah, so luckily I wasn't there. This was in 2021, and I came out of the project in 2023 and 4. I know that the methods are published in a Pineda paper from 2023, but I know what they were doing is basically with the convergence zones, they were actually kind of sampling in a serpentine pattern to cross through. Otherwise, your nets just get all completely gummed up. And then the paratoes, I don't remember exactly how far off the convergence they were, but I think they were still roughly within sight of where they were.
Thank you.
Anyone else?
Hi, thank you. Great talk. I'm curious to know a little more about the metabarcoding results that you had. You mentioned that you didn't find any copepods in the metabarcoding results. Do you have a theory as to why? And then with regards to the fish in the results, do you think that's truly based on diet, or could that have been residual from tow and library prep?
Yeah, so for the copepods, I think that is simply because of the marker choice, and the sampling out of a predator's gut is just difficult. It's always kind of messy. This is one data set I have with metabarcoding and gut contents. My thesis is another data set with some more samples where we did see a lot of copepods within the metabarcoding, and that was 18S barcoding using the blocking primer. So now that we are about to do that here, I'm expecting to see the copepods show back up. So my guess is it was simply competition for primers and other prey just pulling it away from the copepod DNA. In terms of the fish, yeah, I think there is always a question of whether any prey, the sequences you get could be from some kind of environmental contamination or something. I would say with microscopy work that I've done, I have seen fish eggs in larval guts before. And so I would say I feel relatively confident that is in fact a diet constituent, yeah.
Out of curiosity, what reference database did you use when you were doing your analysis?
Yep, so I've used Silva usually, yeah.
Okay.
And then I use the data2 pipeline.
Perfect, thank you.
Unfortunately, we are out of time for questions. Thank you very much, Alex.